Hi guys, welcome back. Thanks for joining me today. This is our first video in a series that I'm going to do on antibiotics, which obviously they're an incredibly important part of medicine. They've made medicine what it is today, save an uncountable number of lives each year, easily stretching into the millions, and also very topical because with the mounting antibiotic resistance growing globally, it's really important that we understand how the current antibiotics that we have work, how to use them effectively, and potentially new targets for new antibiotics as well. Before we can jump into specific antibiotics, I'm going to do a lecture on each kind of some of the broad classes of antibiotics. Before we jump into that though, there is probably something really key that we need to cover about some bacteriology. That's this concept of the peptidoglycan cell wall. So if we skip this for a moment, you'll remember that there's two types. We broadly class bacteria into two types. Let's say the gram-positive or gram-negative. This gram status is based on how they look when we perform a gram stain, and the gram stain is looking at the amount of peptidoglycan within that bacterial cell wall. So gram-positive bacteria, these guys have got a lot of peptidoglycan. So lots of peptidoglycan, and they're a bit simpler. So they'll have this really nice thick peptidoglycan cell wall, and then they'll have their smaller kind of membrane in here, and this is the bacteria in here. So this is outside the bacteria, this is the cell wall, this is the thin inner membrane. So that's what a gram-positive looks like, and the reason I've actually chosen these colors is because on a gram stain, gram-positive bacteria actually come up this really nice purpley color. Gram-negative bacteria, on the other hand, they've got a very thin, let's say a thin peptidoglycan layer, or helps fucking spell, thin peptidoglycan layer, and because of that, they've actually got two membranes. So once again, we've got this little peptidoglycan layer, and we have this inner membrane still, and then we actually have, we'll stay with red, we have an outer membrane here as well. So you've got this thin peptidoglycan layer in between two membranes, so outer membrane and your inner membrane. This outer membrane is important for a number of reasons. It's where LPS comes from, and we'll discuss that in another video. It's not really important at the moment, but it also means that when we do a gram stain, there's actually less peptidoglycan to stay blue, and so basically how we stain for these is we get some bacteria, we add the blue stain, then we wash it out really quickly and quickly add a red stain. In a gram-positive, when we stain the peptidoglycan, there's so much peptidoglycan there, it sucks up a lot of that blue dye that when you quickly wash, not all the blue dye gets washed out, most of it gets stuck in this peptidoglycan. So when you stain it with the weak red dye, there's really not, it's very much overpowered by the blue. Here though, because we've got such a thin peptidoglycan layer, when we add the blue dye, as soon as we start to wash it, it all washes out really quickly. There's not enough peptidoglycan there to hold enough dye, and when we add our counter stain or iodine, it shows up red instead. So that's why we see the two different colors. So you might be wondering why this is important. There's two reasons really. The first is that this peptidoglycan layer is a really good target for antibiotics. It's where our main class, penicillins and beta-lactams, that's where they work. It's really important and a really great target because our cells don't really have anything like peptidoglycan. That's one of these key principles of antibiotics, is that they need to be targeting things that our body doesn't have, because if the antibiotic is targeting a process that our cells have, as well as killing the bacteria, it's going to kill our own cells, and obviously that's really bad, and that's how you get side effects. So targeting the peptidoglycan wall means that we can get these drugs with very few side effects, but with really good efficacy. So that's one reason. The second reason that we need to understand this cell wall concept is because it affects the way some drugs can get into the cell. So some antibiotics that we give work in here, inside the cell, inside the bacteria. And so to do that, they actually need to cross this layer. And because of that, some antibiotics will only work on specific classes of bacteria. So that's another reason why this is really important. If we go back up to here then, I know this is looking overwhelming for now, but let's just focus in on this thing here. This is, if I zoom out again, ignore all this down here for now. What we're focusing on is this. This is the backbone to our peptidoglycan wall. So might be a little confusing at the moment. You don't need to know this in too much detail, but we're trying to get a general appreciation of what's going on here. As a side note, these kind of shortenings here, this stands for N-acetylglucosamine and N-acetylmuramic acid. These are just two of the backbones, the kind of the backbone molecules for peptidoglycan. We then got two phosphates and then a lipid. So it's like a phospholipid kind of structure here. And then you might recognize these from your biochemistry. These are amino acids. Now, something interesting in bacteria, so in all of our cells use L amino acids. And that's based on L-glycine. That's the precursor molecule to do with chirality. So the way glycine rotates light is what dictates how these, what these amino acids are named, whether they're L or D. That's irrelevant for now. You can watch another video on that if you're interested. But the important thing is that most cells only use L amino acids. Bacteria are really cool. They actually alternate L and D amino acids. What that does is you basically means that you're stacking in these tessellating amino acids so they can pack really tightly. Gives the cell wall a lot of rigidity and strength, and so that's a really cool kind of I guess feature of bacterial cell walls. We've also got this thing here, this pentaglycine chain. So coming off our lysine residue here, we have 5 glycine residues, and you will see why that's important in a sec. These precursor kind of building blocks, these are manufactured inside the cell, and then there are specific transporters which are going to move it to outside of the cell where there are enzymes that are involved in actually building up that cell wall. So this is specifically the Staph aureus building block. There are some differences between bacterial species. And if we go out here, this is what cell walls look like. So it's a polymer. So these chains, they continue and they run the whole kind of length around of the cell wall, and they're linked together by these pentaglycine chains. So you can see here, so we've still got an N-acetylglucosamine and an N-acetylmuramic acid. We've still got those amino acids here. You might notice now though that we've only got 4 amino acids, whereas here we have 5. 1, 2, 3, 4, 5. So what's important here is this terminal D-aller chain, this guy right here, that's chopped off by the enzyme that stitches this cell wall together. It's chopped off and attached to the glycine residue that's also, you can see, attached to the pentaglycine here to the lysine. So this is one of our building blocks. If I can kind of circle that, if that makes sense. So that's one of our building blocks right here. And you can see, so our lysine pentaglycine chains going off here. This is our terminal aller, because this one's been chopped off. And then the enzymes linked the glycine chain of another one of the residues so that these residues are now stuck together. So this pentaglycine chain is really important. It acts almost like the cement for the wall. It's linking all the blocks together to build this really strong cell wall. Now, the terminology for the enzyme that does this can get a little confusing. We have 2 names for it. Let me change color. So the enzyme that does this is called either a penicillin binding protein, PBP, or it's called a transpeptidase. These enzymes are one and the same. They're two different names for the same enzyme. So what that enzyme does is it sits outside the cell membrane, in the cell wall, or in what's going to become the cell wall, and it chops off that terminal D-aller and attaches it to the pentaglycine chain of another kind of molecule, another one of these residues. And doing that, it forms these really strong bonds, so that then all these little bricks, these building blocks, are now all linked together really well. So probably the most key thing to take away from this lecture into the next few videos, penicillin binding protein is the same thing as a transpeptidase, and it's the thing that builds the cell wall. I'm emphasizing this because this terminology gets confusing, and I always have students that get confused with it. So PBP, transpeptidase, same thing. They occur naturally within a bacteria. They're not induced by antibiotics or anything like that. They're not synthetic. These are made naturally by the bacteria, and they always have been, in order to make this cell wall. For the rest of the videos, I will try and use the term transpeptidase just to try and avoid some of that confusion and things like that, but you will see penicillin binding protein, or PBP, come up, and I might accidentally slip into that in some of the coming up videos, but I just really want to emphasize that now. That's all for today's video, guys. If you got any feedback or questions, please leave them in the comments below. Anyway, thanks for watching.